Today we are going to discuss about transforming principle. Search for the genetic material. Exactly. Search for genetic material. Genetic material. Search for genetic material. So search for genetic material means which one is a genetic material? DNA na, RNA na, protein na. So which one is a genetic material? For that, some experiments had been conducted by some scientists. उसमें important one, transforming principle. Transformation principle. Transformation. In transformation, the meaning of the transformation, transfer the genetic material from one cell to another cell. Transfer the genetic material from one cell to another cell is called a transformation. A transformation experiments conducted by Frederick Griffith first time. Frederick Griffith in 1928. Here also important. Transformation experiments. Who given the transformation principle first time? The transformation principle first time given by our transformation experiment conducted by Frederick Griffith in 1928. Then the organism, which organism he had been carry his research work means Streptococcus pneumoniae. Streptococcus pneumoniae, which causing pneumonia disease. Streptococcus pneumoniae is nothing but bacterium. This is a bacterium. So for transforming principle, the research work carried by Frederick Griffith in 1928. So he conducted the experiments in Streptococcus pneumoniae bacteria, which causing pneumonia disease. So before going to the experiment, then Streptococcus pneumoniae bacteria on culture plate, it producing two types of colonies. On culture plate, it producing two types of cultures. Two types of colonies are producing on culture plate. S strain and R strain. S means rough strain. S means smooth strain. R means rough strain. So S strain, what is the meaning of S strain, sir? S strain means on culture plate is Streptococcus pneumoniae produce smooth and shiny colonies. Shiny colonies are producing on culture plate. In which one? S strain bacteriums, they producing on culture plate smooth and shiny colonies. R strain bacterium, they produce rough colonies on culture plate. Why sir these are producing smooth and shiny colonies? Because they have mucus polysaccharide coat. Mucus polysaccharide coat is present. That's why they are in a smooth in nature. But here mucus polysaccharide coat is absent. That's why they are rough in nature. Then these S strains are virulent. Their colonies are S1, S2, S3 colonies. These are all pathogenic. S1, S2, S3 are pathogenic or virulent in nature. Pathogenic or virulent in nature means they are causing disease. But here R1, R2, R3 are non-pathogenic in nature. Not causing disease. Non-pathogenic nature. So this is an important one. So Streptococcus pneumoniae bacterium producing two types of colonies. Then one is a S strain, another one is a R strain. Sometimes question, what is the difference between S and R strain? S strains are nothing but these strains are producing smooth and shiny colonies on culture plate. Then R strains are producing rough colonies on culture plate. S strain, why they are shiny and smooth? Because of mucous membrane or polysaccharide coat present. That's why they are virulent or pathogenic in nature. Their colonies or their strains are S1, S2, S3. Then here R strain is a rough colonies, mucus or polysaccharide coat is absent. Then their colonies R1, R2, R3, they are non-pathogenic, not causing any disease. Now, a Frederick Griffith experiment, for his transforming principle, what type of strains are used? Yes, S3 and R2 strains are used. S3 and R2. So this is a neat point of view. So which of the following strains are used for his experiment, transforming principle experiments by Frederick Griffith means S3 and R2. Now, what is the experiment of the transformation principle? Experiment, Griffith experiment. So now for this is the basic for that. Now, Griffith experiment, for example, 5-mark question. Can you briefly explain about the Griffith experiment? 5-mark question. Also, with chance to ask question. 5-mark Griffith experiment. So Griffith experiment means, so first time they take in the smooth strains. What I said, S3 and R2 strains are used for his experiment by Frederick Griffith. So first time, then S strains are, S means smooth na, S strains are injected into mice. So S strains is nothing but smooth strains having polysaccharide coat. So that's why they are pathogenic or virulent nature, causing disease. Then mice is died. Mice is dead. Next, R strain or rough strain injected into mice. So R strain not having polysaccharide coat, it is not having a disease character. So injected into mice, then mice is live. So mice is not died. Then heat killed S strain. Heat killed S strain injected into mice. Third case, heat killed S strain injected into mice. Then mice also, that time also mice is alive. Next, fourth step, heat killed S strain and live R strain. Both are combiningly injected into mice. That condition, mice is died. This is the four experiments, 4 times conducted by Griffith. So from this, what he concluded that, see, at first time, S strain smooth colonies are injected into mice, then mice is died. Kaiku, S strain having polysaccharide coat, then pathogenic nature or virulent, then causing disease. Then R strain, rough strains not having polysaccharide coat, not causing any disease, then injected into mice, then mice is alive in condition. Third case, heat killed S strain. This is a causing disease only, but heat killed S strain. When heat killed S strain, the strain is inactive in condition. But however, when heating, some strains are still active. Keep it that mind. So heat killed S strain injected into mice, then mice is not died. Mice is alive in condition. Kaiko, heat killed S strain. S strain is inactive by heating by heating process. S strains are inactive. However, some strains they become active only. You keep it that mind. Now, fourth case, heat killed S strain, live R strain, combiningly injected into mice. Then when combiningly injected into mice, then mice is died. So in this case, he observed that, how it is possible? In this case, in died mice, he extracted some R cells and some S cell. S strain, live S strains also he obtained that. How it is possible? So he thought that he was given 2 types of postulates. At first time, something S2, some information can transfer from S to R, then R become virulent, pathogenic nature, then causing disease. That's why mice is dying. That is the first postulate. Second time what he thought that, no, or from R, live R na. So from this some information transferred to heat kill S strain. Then S become active. So that's why it is causing disease. That's why mice is died. He said second. So many of them they had confused. But finally he given that however it is from R to S, S to R. Some information is transferred. But he given the finally some information can transfer from live R strain to heat kill S strain. Then S become active and causing disease. But some information can transfer from live R to heat kill S strain. But he given that confirmation. He concluded that some information transfer from live R to heat kill S strain. But he didn't confirm or he didn't given the biochemical nature of the transforming principle. So he given transforming principle. What he was given? Something, some information can transfer from live R to heat killed S. Heat killed S. But something is transferred. But he didn't given the biochemical. That something is what? Protein, DNA, RNA. That is the biochemical nature is not given by Frederick Griffith. So finally Griffith concluded that something is transferred from live R to heat kill S strain, but he didn't confirm the biochemical nature of the transforming principle. For that there is another experiment is there. Now next experiment question. So that is a very important next experiment. Then biochemical confirmation of transforming principle. Biochemical confirmation of transforming principle. Biochemical confirmation of transforming principle. So this is nothing but conducted by three scientists. Avery scientist, then Colin MacLeod, then McCarthy. These three scientists conducted the experiments in 1933 to 1944. Year also important. 1933 to 1944 in the organism Streptococcus pneumonia. Streptococcus pneumonia only. These three scientists carried the research work in Streptococcus pneumonia for giving the confirmation of biochemical nature of transforming principle in the year 1933 to 1944. Even theory also 5-mark question. Can you briefly explain about the biochemical nature confirmation of transforming principle? Biochemical nature confirmation of transforming principle. Can you briefly explain? Sir, can you briefly explain about the confirmation of biochemical nature of transforming principle? Or briefly explain about Avery, MacLeod and McCarthy experiment. Anything, no problem. So, transforming principle given by Frederick Griffith. Confirmation for the transforming principle biochemical nature given by Avery, MacLeod and McCarthy. So these three scientists that biochemical nature is what? DNA, RNA, protein. That is confirmed by these three scientists. So biochemical. So who carried the transforming principle experiments for confirmation biochemical nature means Avery, MacLeod and McCarthy. These three scientists conducted the experiments on transforming principle for confirmation of biochemical nature. Then in the year 1933 to 1944 and in the organism Streptococcus pneumonia only. Streptococcus pneumonia only. Some important 1953 Watson Crick given the double helix structure. Years also important. 1953 XRD, X-ray diffraction data given by Wilkinson Franklin. Then 1950 Erwin Chargaff given the Chargaff's rule. Then Francis Crick, 1958, given the central dogma. Then 1869, nucleon given by Frederick Miescher. These words are very, very important. Just given Frederick Griffith in 1928, transforming principle. In 1933 to 1944, biochemical nature of conformation of a transforming principle by Avery, McLeod and McCarthy. These scientists. These three scientists observed the died mice in third case. So these three scientists extracted the molecules from heat killed extracted the molecule from died mice. What molecules are extracted? Protein extracted, then RNA, protein, RNA, DNA. These three molecules are extracted from the died mice. Then for them respectively they taken three types of enzymes. One is nothing but protease. Second one is RNase. Third one DNase. So from these enzymes, what it's are? So before going to the experiment, protease digests the protein, RNase digests the RNA, DNase enzyme digests the DNA. What is the difference between DNA and DNA? DNA is the genetic material, DNA is the enzyme. DNA is the enzyme which acts on a DNA. So before going to the experiment, these three scientists extracted the molecule from died mice, that is a protein, RNA and DNA. For that, respective three enzymes had been taken, that is a protease, RNase and DNase. Protease digests the protein, RNase digests the RNA, DNase digests the DNA. Now come to experiment, how they were carried the experiment. Then you see here, carried the experiment. Then they are taken same mice, heat killed, same thing, heat killed S strain plus live R strain along with the protease enzyme. So first time heat kill S strain, live R strain along with the protease enzyme. I'll give you protease enzyme. Kaiko, these three scientists, Avery, McLeod, McCarthy, these scientists, they expected that the transformation principle may be protein. So that's why they are taking in first case. So heat kill S strain taken, live R strain taken, along with the protease enzyme injected into mice. Then mice is what happened? Died. Mice is died. So when mice died means the protease enzyme cannot affect this transformation. So transformation takes place. So protease enzyme cannot affect the transformation. Something is transferred from live R to heat killed S strain. So that something is not affected by protease enzyme. So not affected by protease enzyme. So that something is not a protein. Mice is died. So transformation takes place. Here transformation, from here to here transformation is not affected by protease enzyme. So protease enzyme actually works on the protein. So the transformation is from R to S, heat killed S is not a protein. From this they were concluded that the transformation from live R to heat killed S strain is not a protein. Then they doubted on RNA, maybe RNA. So then second, heat killed in second case, they were taken heat killed S strain plus live R strain, then RNase enzyme injected into mice. That condition, that time also mice is died. Then transformation takes place. So from this also, here from something live R strain to heat killed S strain, something is transferred. Then the S is become active and causing disease in mice. Mice is died. So transformation taken place. So the transformation transformation is not affected by RNase enzyme. So the transformation is not affected by RNA enzyme. The transforming material, the transformation material is not a RNA. They were concluded that. So from this case, I repeat that. I revise. See here. From live R to heat kill extent, something is transformed. Something is transformed. Transform means transformation takes place. The transformation material is not affected by RNA enzyme. So that material is not a RNA. So from this concept, from live R to heat kill extent, the material which one is transferred, that's not a RNA, that's not a protein from above two experiments. So they are thought that it is not RNA, it is not a protein, maybe confirmly it is a DNA. For confirmation they go for the experiment. Then heat kill as a strain. In third case, heat kill as a strain. Then live or strain. Then injected along with the DNase enzyme, injected into mice. Then mice is died. So then no transformation. This is very, very important, Hannah. So in this case, heat kill as a strain, live or strain combinedly along with the DNase enzyme injected into mice. Then mice is here not died. Mice is a live in condition. So mice is live in condition. So when mice is live means from here live or strain to the genetic information not transferred to S, not transferred to heat kill S. So S not become active. So it is not causing disease. Means mice is not a died, live in condition. So no transformation. So from live R to heat kill S, no transformation when DNA is enzyme used. So DNA is affect the transformation. DNA is affect the material which is transferred from live R to heat kill S state. So that material is nothing but DNA. So they confirmed that above 3 experiment. The something is transferred from live R to heat kill S strain. The transformation material is not a protein, is not a RNA, it is a DNA. From Avery, MacLeod and McCarthy, these 3 scientists, they confirmed that the material which is a transfer from live R strain to heat kill S strain, which is causing disease and causing for the died of mice, the transformation material, the transformation biochemical nature is DNA. That is confirmed by Avery, MacLeod and McCarthy scientists. I will give this once again, revise. So thoda careful on that. Avery, MacLeod and McCarthy, these 3 scientists conducted the research works in 1933 to 1944 in Streptococcus pneumonia for confirmation for biochemical nature of transforming principle, which were given the Frederick Griffith. Then in this case, they extracted 3 molecules, biomolecules, protein, then DNA and RNA from died mice. In respect to which works on those 3 molecules, that enzymes also were taken, that is protease, RNase and DNase. Protease works on protein, RNase works on RNA, DNase works on DNA. So then when going for the experiment, so how they were taken experiment? Heat kill S strain, live R strain, combiningly then protease along with the protease enzyme injected into mice. When injected into mice, here to here the material transfer, that already we told. So from something live R strain to heat kill S strain, something is transformed. The transformation is not affected by protease enzyme. So transformation takes place. That's why mice is died, because S become active. So from this case, it is not transformation material is not a protein. Next second case, heat kill S strain, live R strain combiningly injected into mice along with the RNase enzyme. So here when RNase enzyme injected along with these 2 cases, then transformation is takes place, because RNA is not affected the transformation material from live R to heat kill S strain. So then transformation is not affected. So we can conclude that from it is also not a RNA, which is not affected by RNA. So from these two cases, above cases, it's not a protein, it's not RNA, it may be DNA. For confirmation, they had taken heat killed S strain, live or strain, combinedly along with the DNA enzyme injected into mice. Then mice is a live in condition, transformation affected, means no transformation. So from live R to heat killed S strain, the material is not transferred. Why not transferred means DNA enzyme is works on this genetic material, works on this transformation material, digest on the transformation material. So that's why no transformation takes place. That's why heat kill S strain is not become active, not causing disease. That's why not died. So from these three cases, Avery, MacLeod, McCarthy, these three scientists confirmed that the transformation principle who were conducted by Frederick Griffith, the biochemical nature of the transformation material from live R to heat killed S strain is 100% DNA. DNA only transfer from live R to heat killed S strain. That was given by Avery, MacLeod and McCarthy. However, some other scientists, biological scientists, they didn't accepted this theory. So for DNA is a genetic material. Is DNA is a genetic material? For that confirmation, again one more experiment came. That is Hershey and Chase transduction experiment that we will discuss in next class.